recognized immediately how important mTOR would be and rapamycin. But I actually started working on mTOR in yeast kind of by accident. So we were really interested in understanding what are the genetics that control longevity. And so we did an unbiased search for new genes that would affect lifespan and happened to find mTOR. And when we made that discovery, I immediately went and looked up everything I could learn about mTOR and found out there's this drug, rapamycin, that's an inhibitor of mTOR. And then we found that we could also increase lifespan with rapamycin. At this point, we were working in yeast, but then it became clear to me, because of the work of others, that this pathway and this drug appeared to affect the biological aging process, not only in yeast, but also across the animal kingdom. And we now know even in mammals like mice and potentially in larger mammals like dogs and people. So I think with that knowledge, I got very interested in trying to understand what the mechanisms are for how rapamycin was affecting the biological aging process. We've studied this in yeast and worms and fruit flies and mice, a little bit in pet dogs, which we may talk about. And I think through all of that, the one thing that has kind of kept me excited about rapamycin as a potential longevity therapeutic is that it always works. And I would say, without question, it is the most robust and reproducible drug, at least from preclinical studies, that we know about today for impacting not only longevity, but to the extent that we can measure various metrics of health span in complex animals, rapamycin also seems to positively impact pretty much every aspect of health span that we measure. So